Welcome back, everyone. Many apologies for the very short break that you've had. I think this proves to us once again that we have too much information that we want to share. But this is the last session of the day, and we're focused on vaccine safety. And this final session of the day has 4 10-minute presentations on different aspects of, first of all, vaccine safety surveillance, and then 1 clinical assessment system that's also engaged in research around vaccine safety. And the presenters will be me, Monika Naus, Julie Bettinger, Naveed Janjua, who is in Halifax today and will join us for the Q&A, but his presentation has been recorded, and then Manish Sadarangani, who you met earlier as well, and the bios are in the program. Okay, so these are my disclosure slides. I have no financial conflicts of interest to declare, and I am a salaried public health physician and professor at the University of British Columbia, and my biases are very much in favor of prevention and public health, even though I do want to, none of you need to be reminded, but sometimes we need to remind the outside world that in public health, we don't just care about the herd, we actually do care about individuals as well, and individual safety is paramount. So the goal of public health immunization programs is to achieve and maintain high levels of vaccine uptake. And this is true whether the goal is disease control or more ambitious like elimination or eradication of a disease. And concern about vaccine safety is a key driver of decision making for the public about their own or their child's immunization. And this is true during the COVID campaign, but it's also true with every survey we've ever done or anybody's ever done around knowledge, attitudes, and beliefs and what drives decision-making. So, paramount is concern about the disease, but equally important is always concern about vaccine safety. And what's shown on this chart is that as disease rates decline after the introduction of a vaccine program, there can be a loss of confidence about vaccine safety and increased concern about vaccine safety. And sometimes that can be reduced when there's a resurgence of the disease because of loss of coverage. And so this is just a cycle that is sort of a classical description of what occurs, but I think it's true to say that vaccine safety is a concern oftentimes right at the start of a program. Many of the diseases against which we have begun to immunize in more recent years are perhaps less frightening to the public, including things like chickenpox, and you all know about chickenpox parties or rotavirus infections. And obviously, as we offer COVID vaccine, the interest in COVID vaccination has also declined as the incidence of this disease has declined. But that's a good thing. That is the goal of our immunization program. And on this slide, there's just a graphic about information on vaccine safety that can be gleaned from a variety of sources. And the components that we're going to talk about are in the post-marketing section here, but very informative to vaccine development is the basic science, because this informs some of the critical aspects of considerations around not only performance for protection, but also the choice of antigen targets. And so a good example of that is the very long time to bring meningococcal B vaccines to the market was partly because they share the organism shares antigens with our nervous system. And there was concern that if we vaccinated, we could see some vaccine safety issues with vaccines with shared antigens to the nervous system. Clinical trials are a key preclinical, so animal studies, but then the human clinical trials are a key source of information. Oftentimes, the size of the trial is designed based on safety considerations. A good example of that are the rotavirus vaccine clinical trials, which were designed for the current vaccines in use based on an observed risk of 1 in 10,000 intussusception with the RotaShield vaccine, which had been withdrawn from the US market in the late 90s and had never been introduced in Canada. Clinical trials can also inform us about potential signals warranting focus in post-marketing safety surveillance. So in both of the mRNA vaccine trials, for instance, Bell's palsies had been observed in both placebo and trial participants, but for one of the vaccines at a slightly higher frequency in vaccine participants, and this was flagged as a potential signal warranting post-marketing safety surveillance. In post-marketing safety surveillance, when findings occur, product monograph information is updated. This can take a little while, but we're a part of a global system, and our regulator, Health Canada, shares information and obtains information from regulators working all over the world, including the US, of course, and the European regulatory process. And so oftentimes they're aware about considerations that are being made elsewhere based on what's been seen in post-marketing safety surveillance, perhaps before we even become aware of it from our own systems. And then continued use of a vaccine or recommendation for use, such as have occurred with the adenovirus vector vaccines for COVID, or vaccine withdrawals, which are extremely rare, can also come from post-marketing safety surveillance. So during the H1N1 pandemic, for instance, a particular lot of vaccine was withdrawn because of an observed risk of anaphylaxis of 1 in 10,000. And we do have preferential use recommendations now around both the adenovirus vector vaccines and the mRNA vaccines because of observation of thrombosis, thrombocytopenia syndrome, and myocarditis. Both passive and active surveillance largely assess individuals in cell A of the table, even though I know that in the Canvas system, there have been attempts to obtain information about the incidence of adverse events or health events in individuals who are not vaccinated. So that's sort of a control population. And so when we think that we are observing a signal in the passive surveillance data, it's important to assess the veracity of that signal using additional information sources. One of those is the occurrence of a particular event in the population at large. So for instance, baseline information on the occurrence of events like Guillain-Barré syndrome or Bell's palsy, sudden unexpected death, those have all now been obtained to support the surveillance in British Columbia using administrative data so that for a particular age group or sex, we know what the frequency of that event is in the population in a period of time, because it's inevitable that in a mass campaign such as we've just undergone, there will be unusual events occurring or serious events occurring in a population that we need to be able to put into context of what the expected rates in the population are for the period of time. Use of linked data that contain both information about vaccine receipt and health conditions and their temporal association are also useful to identify whether an association exists to vaccine receipt. And Dr. Naveed Janjua will describe how the immunization registry data has been linked to administrative data in British Columbia to study the association of specific events, especially myocarditis. And then analytic studies such as case control are ultimately required to verify potential signals or associations, as these also allow for the collection of additional data such as risk factors other than the vaccine, in order to isolate the vaccine receipt as the key risk factor for having experienced that particular adverse event. And then clinical investigations to elucidate the nature of an event and its occurrence require a different type of strategy. And the special immunization clinic that Dr. Sadarangani is part of is a group that can do things like revaccination studies to see whether individuals who've experienced a particular event experience it again following receipt of subsequent doses. So In British Columbia, as in all other jurisdictions of Canada, the passive surveillance system is based on reporting by healthcare professionals. Since 2019, in our province, it's been under a regulatory framework, and the key difference that that's made is that we no longer need consent from individuals to have their event reported. It hasn't really made a big difference in terms of the number of events that we receive. And unlike the US system, where there can be direct from public reporting, we rely on healthcare professional reporting to the medical health officer of health, and in turn to the province and to the federal government. We also have emphasized the reporting of serious adverse events, including those that are life-threatening, resulting in hospitalization, requiring urgent medical care. And in the readiness for the 2009 pandemic campaign, using adjuvanted vaccines, for which we knew that local reaction rates were going to be quite high, with prolonged pain, redness at the injection site, we removed a couple of codes from the then reporting system in order to de-emphasize the reporting of local adverse events, because these do not result in contraindications and are not really what we're interested in. We are interested in serious events. During the current COVID campaign, and on the advice of the BC Immunization Committee Vaccine Safety Working Group, we developed a 2-page reporting form to make it easier for healthcare professionals to report, because the apparently 7-page form was bringing fear into the hearts of reporters. And so that's available now, even though it may require some follow-up by public health to obtain additional information. What the passive surveillance data allows us to do is to basically look at person, place, and time. So we can look at the descriptive epidemiology of the events. And on this chart, it shows the events reported in children under 2 in British Columbia. And the 2 green peaks that you see there are fever and rash reported in association with the 12-month vaccines that are given. So obviously that's MMR associated, and those events are far less frequent after the 2nd dose, when most children are already immune from the 1st dose and don't experience these. And on this slide, on the left side, I've got an image from Barbara Law, the former chief of vaccine safety, who, I think this is a photograph from the 60s at the old Laboratory Center for Disease Control of Ed Napke, who was then the head of drug and vaccine safety. And he had a color-coded pigeonhole system for signal detection, potential signal detection in the reporting of adverse events. And we've come a little way since then, but we do have a statistically based algorithm that looks at expected and observed frequencies of events and spits out a signal for us on a weekly basis. And Hadi Dilati, who's here today, is the epidemiologist who, along with the surveillance analyst, reviews those on a weekly basis. During the COVID campaign, we've been producing reports on a weekly basis. Again, it's been Hadi's task to do that now, but Kyle Noftel was our epidemiologist at the start of the campaign, and these are now produced monthly and are posted at this link on our website. And I won't go into these, but we have identified through this system the 6 thrombosis and thrombocytopenia events related to the adenovirus vector vaccines, and as of our most recent report, 231 events of peri and myocarditis. And what's interesting on this slide, and you'll have felt it in the regions, if you're part of the adverse event reporting system, is that we had a lot of reporting in the early parts of the campaign, and you can see the number of doses administered in the red line here, and you can see that even though we've had several peaks of vaccination, we've had far smaller event reporting rates subsequent to that very first part of the campaign. And this is a phenomenon actually observed for introduction of drugs into the market, and it's called the Weber phenomenon, whereby as we become more familiar with the expected side effects, including providers, there is a decline in adverse event reporting that occurs. Finally, I'll just like to touch on the fact that we are trying to introduce a more standard process to a serious event review. This is an algorithm that's developed by the World Health Organization, but very similar to the type of system that was used formerly by the National Advisory Committee on Causality Assessment, which sunset after the 2009 pandemic. And we've had several discussions with medical health officers in the province about trying to build this into at least a select group of serious adverse events in order to standardize the review that to some extent they need to do anyway when they're making a decision or a recommendation about whether it's safe for the individual to proceed to vaccination or whether further investigations need to be done. And finally, I just want to flag that all of the reporting that you do is part of a much bigger system because we are part of a global vaccine safety surveillance network. The vaccines that we use in this country are used in many other countries as well. As time has gone on, there are fewer manufacturers for the routine childhood vaccines, for instance, maybe one or two that supply the entire globe with vaccines. And so it's important to be part of that network. And one of the Global Vaccine Data Network at the bottom right there, run out of New Zealand and funded by CDC Atlanta, has 2 participating sites from Canada, including Ontario and British Columbia. And up in the top right corner, I have a photograph of the holy grail of vaccine safety, which is when I started my job at BCCDC many years ago, Bob Brunham, who was then the medical director of the center, said that genetics will revolutionize the practice of public health in the next 10 years. And so that should have happened in like 2011. And one of the things that it could revolutionize, if we were smart enough to do this, is to identify those rare individuals in the population who may experience that Guillain-Barré syndrome or myocarditis event after receipt of specific vaccine products. And I don't know whether we will, if you haven't seen this movie, you should. But I don't know whether we will ever get to that for vaccines, but it is the Holy Grail. Thank you. So.
Thank you. So we're doing, as was said, kind of a tag team approach. So let's see if I can, yes. So I'm going to speak a bit, oh my gosh, I think you saw my disclosures earlier. I am a member of the National Advisory Committee on Immunization and the BC Immunization Committee, which are probably the 2 relevant things. And I don't have any affiliations with pharmaceutical, medical device, or communications organizations. I'm at BC Children's Hospital in the Vaccine Evaluation Center there. So I'm going to talk today about active and sentinel surveillance, which can be sort of considered a subgroup of active surveillance that we use for monitoring and evaluating vaccine safety in Canada. And I'm hoping if you didn't get it from Monika's talk, you'll get it from mine in terms of what a vaccine safety signal is. So active surveillance is essentially, Monika talked about passive surveillance and described that pretty well, I think, whereas active surveillance is slightly different in that we're doing outreach to detect cases or to stimulate case reporting. So instead of sort of the local public health unit waiting for healthcare providers to tell you about something, we actually have people going out and looking for these events. And so this can involve searching hospital records, laboratory reports, discharge summaries. There's all sorts of places you can look for this information. When we do it in sort of a selected group, or oftentimes it's usually set out sort of in a geographic location, that would be what we would call sentinel surveillance. And ideally, the group you pick is representing the entire population for this to work really well. And you also should really be using sort of standard case definitions and protocols so that if you have several sentinel sites, they're all doing it the same way, and you end up getting comparable data at the end that you can combine and then look for signals with that. So I think I'm not going to spend time on this. Well, maybe I will quickly, because I don't know if Monika went through it here. But essentially, when we're looking for a vaccine safety signal, this would be reports of a specific type of adverse event. So myocarditis is a great example, occurring after a specific vaccine that suggests a new or potentially causal association or change. So these can be clusters in a region, a new type of event that we haven't seen. Okay, they are advancing on their own. So reports of a new type, an unexpected increase in something that we're familiar with, like GBS, as we've already mentioned. Or so oftentimes we may hear something from another jurisdiction or the vaccine manufacturer about a specific lot or a specific product, and that might prompt us to start looking to see whether we're seeing anything in our symptoms or in our systems. And I think what's important to sort of recognize is when we're looking for these signals, we're not able to establish causality, and Monika did touch on this. We're really looking for temporal events. So it's really, did this occur after the vaccine? I can't tell you whether or not the vaccine caused that event. I'm just going to be able to tell you, yes, it occurred after the vaccine. That causation piece has to be evaluated differently, and our active surveillance is not going to really be able to tell us anything around the cause of it. She talked about background rates, so I'm not going to go into that. But we do have sort of two active surveillance systems that we use in Canada. So this is hold on, yeah. So we've got IMPACT, which is an active sentinel surveillance system of selected adverse events leading to pediatric hospitalizations, and this feeds into the Canadian Adverse Event Following Immunization Surveillance System, which is our passive system. So we've got our active system in our pediatric hospitals set up to find serious and severe events and feed those up into the passive system. So it's sort of creating some enhanced surveillance for our passive system. And then we've got the Canadian National Vaccine Safety Network, which I lead, or CANVAS. And this is a slight difference from our other reporting systems in that it's participant reporting. So we actually ask the individuals who have received the vaccines to tell us about any health events that are occurring in that first week after vaccination. And then depending on what we're looking at, sometimes we have longer follow-ups. We did that with COVID, where we followed up people at sort of a month and then 6 months. And this is slightly different than what you're going to get from the passive or active systems, but it is going to give you the ability to really look for signals. So to tell you a little bit more about IMPACT, or the Canadian Immunization Monitoring Program Active, this is in 12 tertiary care pediatric centers across Canada. It's been in place since 1991. You can see the little dots on there for where all the centers are located. The network covers over 90% of the tertiary care beds in Canada, and we get referrals from all the provinces and territories. So it's a pretty comprehensive system for Canada. And the way this works, given that it's active surveillance, is we actually have nurses in each of the IMPACT centers that will scan admission lists on a daily basis to look for the conditions that we have under surveillance. And then what we'll do locally is make sure that anything we find that's reportable goes into the local public health and then gets referred into the passive surveillance that way. And we also send them directly to the public health agency. And then when both those reports get to the public health agency, they deduplicate it, so we're not counting things twice. But that way, you've kind of got really quickly reporting at a federal level for some of the more serious events. And in 2021, we did start monitoring for some COVID-specific APHEs. And so this here is just showing you the list of adverse event targets that we monitor. I won't go through all of them. You can read it. And the ones that we added for COVID were myocarditis, pericarditis, and then multi-inflammatory syndrome in children, or you may have heard this referred to as MIS-C. So what does IMPACT do? If you look here on the slide, I'm not sure what that would be on your left. So the blue bars are what are coming into the passive surveillance system, and you can see the sort of orange down there, the really small is what IMPACT puts in if you look at the total number of reports. However, if we look at the serious reports that are coming into the passive system in that next graph, you can see that IMPACT is accounting for more than half of all the serious reports coming into the system, and of the about 70 to 90% of all the neurologic APHEs that get reported. So in terms of what it was set out to do, it's doing its job well. And then there's some highlights here. There's also a publication list. The link is down there at the bottom. So if you're interested in any more specific information on any of these topics, I would refer you to that. But you can kind of see we've looked at a lot of vaccine safety over 30 years that impact has been around. There are challenges and limitations, as there are with everything. And so I think with any sort of active surveillance system that's in a hospital, we're only going to be catching the tip of the iceberg, right? I mean, we're set up to really capture the serious, severe events that are coming in. We're doing that well, but as you saw from the other graph, we're not capturing everything. We've heard this from some of the other speakers today, the challenges around immunization history and tracking that down so that you know if that GBS case that was admitted to hospital had a flu vaccine or not. And without having kind of a national immunization registry, it really, really is hard to do that sometimes. And sadly, this information is almost never completely captured in the chart. At most, it might say the child is up to date in their immunizations. And so you can't really do much with that if you're really trying to figure out, you know, did this event occur after a vaccination, right? Our case identification is really dependent on local testing and admitting practices, and it's quite labor intensive. It's active surveillance. The nurses are screening over 6,000 admissions annually to find, you know, maybe 100 cases. So the next one I want to touch on in my remaining time is Canvas, and we did it for both COVID and influenza surveillance, and we've got over 1 million participants that have enrolled in this, and we're in a number of provinces and territories across Canada. We collect information on the age group, vaccine type, health status, and for COVID, whether or not they'd had a previous COVID infection. The online survey that we send out is capturing health events in the first week after vaccination, and we really are looking for what we have defined as severe events. So these are events that cause work or school absenteeism, prevent daily activities, or require a medical visit. What's unique with Canvas that we don't have with some of our other surveillance systems is that we actually have a control group of unvaccinated individuals. And that allows us to then really be able to detect signals and to have that background rate. And so we can see if any of our events are occurring more frequently in our vaccinated group than our control group. And the controls fill out the exact same survey for everything except injection site reactions. So this is just kind of a summary of what we've done during COVID. We were providing weekly result summaries to federal and provincial public health. We had various signals that we investigated during the campaign. We looked at the safety of COVID in adults. We checked on changes in menstruation. We've looked at the safety of vaccines in pregnant people. And looked at the safety of vaccines in people who've been previously infected with COVID. And some of the articles are down there if you want the details on that. Again, strengths and limitations of this system. It's real-time surveillance. We were hearing about the menstrual changes before it was really popping up anywhere else. It was quite interesting, and we added that as one of the symptoms that we started tracking sort of formally. It's relatively low cost. You know, we're not having to pay nurses to look for these things, and it's really adaptable. We've used it for influenza vaccines. We've used it in Quebec when they rolled out their meningococcal B program, and we've used it with COVID. So it's, you know, it works very well, particularly for vaccines that are given in a mass immunization setting. So the challenges are, it's patient self-report. So as you all know, I'm sure you're aware of the challenges with that. We're not getting it from a chart, we're not getting it from the physician. Depending on who the population is, we may have low power to detect rare outcomes if we don't have, you know, if it's a 1 in 3 million event and we've only got 1 million people enrolled, we're not really powered to find those types of events. And then I think really importantly, our respondents are likely not always representative of the general population. And I think I'm out of time, so I'm gonna skip these. Okay, so I think hopefully what you've seen from sort of Monika Mai's presentation is that we really closely monitor vaccine safety here in Canada. Active surveillance really enhances our passive surveillance system and can provide us with a lot more information. But importantly, both of these systems are measuring temporal events. They can't tell you whether or not they were caused by the vaccine. It's just our first starting place to then do some other studies to try to figure out whether or not there's a real association. So thanks, I'll stop there.
Good afternoon. I will be talking about vaccine safety, what we can learn from administrative data linkages. Before I start, I would like to start with the acknowledgement that this work was carried out on the traditional, ancestral, and unceded territory of Coast Salish people, including territories of Musqueam, Squamish, and Tsleil-Waututh nations. Today, I'm joining from the city of Halifax, located in Macmahon, the ancestral and unceded territory of Mi'kmaq people. The people of Mi'kmaq Nation have lived on this territory for millennia, and we acknowledge them as the past, present, and future caretaker of this land. I would also like to acknowledge that this work was carried under BC COVID-19 cohort vaccine safety working group, and notably led by Dr. Zahima Naveed, who is a postdoc with our team. These are my disclosures. Data, when we seek healthcare, these data are captured in healthcare data sets, and when these data sets are integrated together, it could create a longitudinal medical history of each individual's, including medical visits, emergency room visits, hospitalization, prescriptions, etc. So this could act as a surveillance system. It could be used to assess gaps in care, as we have developed Care Cascade for hepatitis C to monitor program effectiveness. And it could be used to assess intervention effectiveness, as we have done for the effectiveness evaluation for hepatitis C treatments. And it could be used for pharmacovigilance. And in other jurisdictions, these data sets were used for vaccine safety monitoring. And here, we have not been doing that. But at the beginning of COVID-19 pandemic, BCCDC, Ministry of Health, and Data Analytics Reporting and Evaluation team at BHSA came together to create an integrated data set, integrating data on COVID-19 case surveillance, lab testing data, hospitalization census, vaccination, and integrating that with various administrative data sets, including MSP, hospitalization, prescriptions, and various demographics data. Some of these data sets are updated on daily basis. Some are updated on weekly and monthly basis, and that enables setting up system for monitoring vaccine safety. And that's where we started to assess potential use of this system for vaccine safety monitoring. We did various assessments, but today I will focus on myocarditis related studies that we did. We did estimate background rates for various conditions of interest, but here I will focus on myocarditis. We did analysis looking at observed versus expected rates. We did comparative risk of assessment of comparative risk of myocarditis following second dose of Pfizer, BioNTech, and Moderna Spikevax vaccine. We did assessment of myocarditis following mRNA booster dose vaccine as well. So I will start with observed versus expected rate of myocarditis. So this was an observational study using data from BC COVID-19 cohort from December 15, 2020, when vaccine became available, to March 10, 2022. And primary exposure was any dose of an mRNA vaccine against SARS-CoV-2. Primary outcome was hospitalization, emergency room visits for myocarditis or myopericarditis within 7 and 21 days post-vaccination. We computed myocarditis rates per 100,000 vaccine doses, and we computed expected rates from 2019, and then we computed observed to expected ratios using these observed and expected rates. This slide shows the overview of results. We found 99 incident cases of myocarditis within 7 days, resulting in observed versus expected ratio of 14.8. And the slide presents overall rate and by various demographics. And we could see that the rates were higher among males, among age group 12 to 17, 18 to 29, following Moderna Spikevax, and following the second dose. And the observed versus expected ratios followed the similar trajectory as the rates. So this second slide from this study dig deeper into these results, where we stratified these results by age, sex, and vaccine product. And we see that the males who were between ages 18 to 29 and received Moderna Spikevax, presented by orange bars, had higher rates compared to other groups. These were followed by age group 30 to 39, and then the rates were not that high in remaining age groups or combinations. Just to summarize, overall absolute rates of myocarditis following mRNA vaccine were low. We observed highest observed to expected ratio after second dose among males 18 to 29 years, and the second highest rate observed, and the highest rate observed among males 18 to 29 years who received Moderna Spikevax vaccine. These findings supported the use of preferential use of Pfizer-BioNTech vaccine over Moderna Spikevax for people ages 18 to 29 years. The second study was comparative risk of myocarditis following second dose of Pfizer-BioNTech and mRNA 1273 vaccine from Moderna. And this was a head-to-head comparison. As we see that in the previous analysis, we saw a signal of higher rates following Moderna Spikevax, but that wasn't a head-to-head comparison. So here we did head-to-head comparison of those individuals who received Moderna vaccine against the Pfizer vaccine using the data from BC COVID-19 cohort. In this analysis, study population included these people who received second dose of an mRNA vaccine and the diagnosis of myocarditis, pericarditis, and myopericarditis during an hospitalization or emergency room visit within 21 days of second dose. So this slide provides an overview of results overall between December 15, 2020 and September 9, 2021, the study period. We had about 3.2 million recipients of second dose. And then out of these, about 3 million fulfilled the eligibility criteria, and about 2.2 million received Pfizer-BioNTech, and about 872,000 received Moderna Spikevax vaccine. Within Pfizer-BioNTech, there were 28 events of myocarditis, resulting in the rate of 12.6. Among the recipients of Moderna Spikevax, there were 31 events, resulting in the rate of 35.6. When we looked at the rates stratified by age and sex and product, we found that rates were much higher among males 18 to 29 years, followed by 30 to 39 years among the recipients of Moderna Spikevax vaccine. Just for the y-axis scale, so this is rate per million doses. This slide presents the overall and stratified logistic regression analysis. And at the bottom of this graph where we start, the overall rate, results comparing odds of myocarditis following Moderna compared to Pfizer-BioNTech vaccine, and you could see that the odds ratio was about 2.5. Similar results, slightly higher for males. Results were not significantly different for females, but when we look at age group 18 to 39 years, odds ratio was higher than 5. And there were no difference in age groups greater than 40 years. To summarize, this was head-to-head comparison of two vaccine, Pfizer-BioNTech and Moderna Spikevax. This also showed that people who received Moderna Spikevax vaccine were 2 to 3 times more likely to experience myocarditis than people who received Pfizer-BioNTech. And association was stronger for men in ages 18 to 39 years. Then following this, we did assessment of myocarditis following mRNA COVID-19 booster vaccination among adult recipients to estimate the rate. So the overall objective for this analysis was to estimate the rate of myocarditis following COVID-19 booster vaccination. And of course, like the exposure was booster dose of an mRNA vaccine, and we looked at myocarditis by pericarditis within 7 days of the receipt of a booster dose vaccine. And we compared these rates with the second dose. We computed myocarditis rate, we computed rate ratios and risk difference between second and the booster dose vaccine. This slide provides an overview of results. We could see that in the maroon reddish bars, which present results for booster dose, overall rate was 6.08 per 100,000 doses, which was much lower than the second dose. And when we look at the across vaccine products, rate was similarly within males, within females. And then when we look at males 18 to 29 years, the rate were slightly higher among recipients of the Moderna Spikevax compared to Pfizer-BioNTech, but we should be cautious about this because number of events are very small and we cannot draw any conclusions there. So overall, we could say that the link administrative data sets are important tool for vaccine safety surveillance. We did it here for COVID-19 vaccines, and I presented data for myocarditis, but it could be used for other outcomes following COVID-19 vaccines and other vaccines as well. Related to myocarditis, myocarditis following mRNA vaccine is rare event. People who receive Moderna Spikevax were 2 to 3 times more likely to experience myocarditis than people who receive Pfizer-BioNTech following second dose. The rate and association between Moderna Spikevax and myocarditis was strongest among males 18 to 29 years following second dose. Rate of myocarditis following booster dose was lower than following the second dose, and there was no difference between Moderna Spikevax and Pfizer-BioNTech vaccine. I would like to acknowledge teams within BHSA, Data Analytics Services, and DARE who supported creation of the cohort and data stewards and funding from CHR, Canadian Immunity Task Force, and Public Health Agency of Canada. Thank you very much for your attention.
Okay, I just want to start thanking Monika and the organizers for inviting me to join this discussion around vaccine safety. And as Monika mentioned earlier, I'm going to talk specifically around the Special Immunization Clinic Network. Okay, so I just want to acknowledge where I work at BC Children's Hospital, and where we are is on the traditional ancestral unceded territory of the Coast Salish people, including the Musqueam, Squamish, and Tsleil-Waututh nations, and I feel very privileged to be able to work and live and play on these lands. These are my disclosures, so I have some salary support from the hospital and from the Michael Smith Foundation for Health Research. I've worked on projects funded by multiple vaccine manufacturers, but all the funds go to UBC. Okay, and then that's the medications. Okay, so this was the brief that Monika gave me when she first emailed me, was to be familiar with the contribution of the special immunization clinics to research in vaccine safety, including allergic events with COVID-19 vaccines. So I think this is a sort of similar concept that Monika also had on one of her slides, that vaccine safety really spans, from the molecular level all the way to the population level. And what I'm going to focus on is around individual patients and providers, and then talk a little bit about how we're moving into maybe, I'm not going to commit to 10-year timeline, but maybe into the future. So the Special Immunization Clinic Network, so it's led by Karina Top, who's been based in Halifax, and is a national team of expert clinicians, including specialists. Initially, it was infectious diseases and allergy, but it's really expanded during the pandemic to include immunology, hematology, oncology, transplant specialists, neurologists, and I guess as different safety signals come up, it will expand in different directions in the future, I'm sure. So we do standard patient assessments in our clinics. So I'm at the clinic at BC Children's Hospital with Dr. Jane Finley and Annie, who you heard from earlier as well. And we've really expanded over the pandemic. There's a lot of, and I really want to acknowledge the huge amount of support we've had from the adult physicians in BC over the last 3 years to help supporting assessment of adults. So we look at patients who've had previous adverse events, and also those people who've got comorbidities that may affect their vaccine response that need evaluation before they get vaccinated for some vaccines. We have a number of ongoing studies, including management of patients who have contraindication to vaccination and those with APHEs, optimizing varicella immunization after transplant, and I'll just talk very briefly about that at the end, and also the safety of rotavirus vaccination in infants born to people on biologics or monoclonal antibodies during pregnancy, and I'll very briefly touch on that as well. So this is a study that's already been published. This is pre-pandemic data looking at this is mostly children. There's very few adults in this study looking at the ones who are referred to us with previous APHE. And you can see that the vast majority of those referred to our clinic, so 87% were recommended to be revaccinated. And then of the ones that we could follow and find the records, which is not always possible, and there's been lots of conversation about that, but about 2/3 of them, we could track their records of revaccination, and 90% of them did not have any recurrence of their APHE, and 10% did. And I just want to very briefly, I said the publication's there and you can look at it, but in terms of the outcome, the highest rates of recurrence were in those who had large local reactions to start off with, so that was about 30%, and then about 20% in the other injection site reactions, and very small numbers in the other groups. And for the most part, the recurrence was either milder or the same severity as the original APHE. As part of a sub-analysis of this study, there was 92 participants who had suspected allergy, and they had skin prick testing and were revaccinated as well. And the negative predictive value of the skin testing, so if you had a negative skin test, the likelihood of you having no APHE was almost 100%. So a negative skin test was very helpful. Positive skin test is a bit more complicated. So then looking at the COVID work that we've done, so this is unpublished data, and this is an analysis that was done by Tiffany Fitzpatrick, and Karina Top has kindly shared the slides with me to share with you today. So we had, in the end, 318 participants, or this is not in the end, this is a previous analysis, so 318 enrolled. Now this is mostly adults, as you can see on the right-hand side there, and so this analysis has been done a bit earlier in the pandemic, and so that sort of reflects where the vaccine rollout was. And you can see in those at the bottom, so just over 100 had some sort of hypersensitivity or allergic-like reaction, just over 70 with acute cardiac events. I think currently now we have about just over 100 who've been referred to the network with acute cardiac events, and then 44 with neurologic events, 31 with hematologic disorders, and then a sort of slight mixed bag of other things. In terms of the impact and seriousness of the APHEs, and this is really relying on that, how that was subjectively determined by the patients. For the hypersensitivity reactions, they're mostly rated as low to moderate in terms of the impact. But as you can see with the other more significant events, like the neurologic events and the cardiac events and the hematologic disorders, there's a bit more of a distribution across this severity spectrum. And then in terms of the revaccination, so this is all the COVID data. So so the darker bars show how many were revaccinated, and the lighter bars show then how many had a recurrence. So you can see with the hypersensitivity, we had around 100 revaccinated, and then about 15 had a recurrence of the APHE. And of the others, the number of revaccinations was lower, one, because the number of referrals was lower, but also, especially with a relatively new vaccine, there was a little bit, I would say, I hesitate to use the word hesitancy, but there was a little bit more caution in terms of recommending revaccination when people have had really serious events. But you can see that the ones that were revaccinated, the recurrence rates were really low. So in terms of future and ongoing specific analyses from these, so the allergic-like events, I really wanted to share that with you today. They're being presented at other meetings this month, so they're not yet public. So unfortunately, this meeting is about a month too early to be able to share that data. Some of you will have seen it in other forums confidentially, so I'm sorry that I can't share that with you, with the whole rest of you. The cardiac events, so that analysis is being led by one of the physicians in Toronto. And so I said we have now around 100 events, mostly myocarditis, also some pericarditis, and some others that are just sort of chest pain without a specific diagnosis, so they don't meet the criteria for either myocarditis or pericarditis, but we have a lot of data on those as well. And the neurologic events as well, that's sort of a little bit more early in the analysis. It's a smaller group, so trying to look at the revaccination outcomes from those as well. And just with the allergic-like events, all I'll say is that we're still, it's very similar to the previous data, so I think we're still finding similar outcomes for the COVID data as the other stuff. I just did want to talk a little bit about the other work that we do as well. So some of you may be familiar with these guidelines that were published in 2019, recommending groups of children who have had solid organ transplant who were deemed to be able to safely receive varicella vaccine. So officially the guidance is still that it's contraindicated for these children, but this is a traffic light system that was developed, and essentially the green was if they meet certain criteria, so depending on how far they are from their transplant, how much immunosuppressive therapy they're having, and other sort of immunologic parameters, that it's safe to go ahead and vaccinate these children with live varicella vaccines. And then we've been following them up for safety, but also looking at their immune response after vaccination. Then there's the sort of orange group in the middle where they're a little bit more immunosuppressed, and we do a little bit more immunologic workup on them before deciding whether or not to vaccinate them. And I can tell you that most of them, when the ones that we've had, so we've had about 15 kids in BC, most of them we've gone ahead and vaccinated them, and the red is these are ones that shouldn't be vaccinated, and the only group from there that I want to pull out is the heart transplant group, and the reason they were put into the red for this guideline was because there was no data, but on expert opinion across the network, we have actually been vaccinating some of those children after cardiac transplant as well with live varicella vaccine. And so there's a manuscript, this data are being analyzed, and we've been over the last 12 to 18 months administering varicella vaccine in our clinic to these children, and we're continuing to do so. And so I think the safety and the immunogenicity data have been reassuring up to now, and hopefully that will be published. And part of that also includes a sort of qualitative study where we've done interviews with the providers, so these transplant physicians and the nurse practitioners who've spent their whole careers telling these children not to get these live vaccines, but also with the parents who've also spent sometimes many years being told, don't give your kids these vaccines, to just gauge their experience as well. Okay, and then this is now stalled. It's died. Sorry. So this is the study that we're looking at. So rotavirus vaccine after in utero exposure to biologics. Many of you be aware these very potent immunomodulatory drugs are being used more and more in adults. And overall, for pregnant people, it's better for them to be on treatment that suppresses their chronic disease rather than not being on the treatment. But that leads to transfer of these drugs across the placenta into the infant that then essentially temporarily immunosuppresses the infant. And so depending on which guideline you look at, rotavirus vaccine, which is the only live vaccine we routinely give, is relatively contraindicated, and we've been working through these these different groups, and you can see here this sort of spider diagram is because there's a lot of different drugs, and they're all a little bit different, and they have their own considerations. So we've got data on about 100 kids now that have received rotavirus vaccine in this context, and these data will be presented at the Canadian Immunization Conference in Ottawa next month, for those of you who are going. So just to come on to the future, just very briefly, so Karina Top, I said, who has sort of been leading the Special Immunization Clinic Network for a long time now, is now leading this global group, which is called INSIS, or the International Network of Special Immunization Services. And part of that includes doing what we've been doing already, linking with other similar networks in other countries, but also starting to collect specimens from these individuals to look at the genetic makeup or to look at specific biomarkers, including in blood or saliva, that you could measure at the time of vaccination to maybe predict who's going to have an AFI or not. And maybe that will help us one day get to where Monika thought well, maybe, I don't know if you thought, but where it was predicted that we would have been in 2011. And right now, we're actually, so the individuals who've had the cardiac events after COVID vaccine, we've been collecting samples, saliva and blood samples from those individuals, but also from controls to be able to start to look at some of these factors in this international network. So that's potentially exciting things to come. Yeah, I think that was my last slide. So there's a huge group of investigators and co-investigators across all the sites. As I said, we've had a real explosion of the number of people in BC. You can see the long list there of different physicians in BC who have supported this work, and I think that's reflected. So the data of the 300-odd patients that I showed you, around 70 of those actually come from BC. So we've been very prominent in the network, which has been really exciting. And then there's a huge team of nurses and coordinators as well who've supported the network. So I'll end there. Thank you. We're going? Okay, we're going to sit down.
So we will take a few minutes for questions. And I believe that we're able to get Dr. Janjua from Halifax onto the line as well. Is he there? Yeah, there he is. Hi, Naveed. And we won't need the full half hour for wrap-up, so you'll still get out of here on time and have a chance to reconnect with your colleagues who are at the meeting. So I guess I jinxed it by saying at the outset that people always get up to the microphone at these meetings, but maybe this is the first meeting you've been to in 3 years, and we have most of our questions have come in on Slido. But the floor is open for questions. Do we have any Slido questions?
Yes. Some people ask about long-term safety of vaccines. How does the current safety data contribute to understanding of long-term safety?
I put this question in because this is a question that the public asks. How do I know that in 50 years from now, I won't get rheumatoid arthritis or some such thing from this vaccine? So,
well, I'll start. I mean, when I get asked that question, I always say, well, we don't. So, I mean, honestly, like we can't say that something that, a cancer that develops 50 years down the road in someone or rheumatoid arthritis, is that related to the measles vaccine they got at 12 months of age? So, I say that sort of flippantly, we can't tell, but there are, certainly in our surveillance systems as they're set up, we can't tell that. They're not set up to do that. As I sort of said, we're measuring temporal events, and something that's 50 years later, the systems aren't going to flag that, right? That's not what they're looking for. But I think some of the other things that we've talked about, the administrative databases, would be a phenomenal place to actually look for some of that information. And in some countries, the Scandinavian countries, Denmark, they've got administrative data on their entire populations, and they're very well linked, and they're very well positioned to look at these types of questions. And in fact, they have done that. So I think there are other methods that we can use to sort of try to tease that out. And maybe I'll stop and let Naveed address that a little bit too. But certainly in terms of our current surveillance, no, that's not what it's set up to do.
Yeah. And I think this is where basic science and understanding of causation and risk factors for development of some of these things also, I think, are very useful. But I agree that our current surveillance systems are not set up for that. Naveed, I think one of the questions that I thought people might be interested in is the COVID cohort. And in fact, there are probably more than one silver lining of COVID, but one silver lining of COVID has been this establishment of these linked data sets to which we've never had access before at BCCDC. And one of the questions and one of the hopes for the future is that we will have access to these types of data in order to conduct the kind of work that we do and that needs to be done for all vaccines, not only for pandemic vaccines. And I wonder whether you can inform that, Naveed.
Yeah, so the administrative data sets that we have now, so they are updated on regular basis, on daily or weekly or monthly basis. And we have an agreement with the ministry that these will be an ongoing feed for us. And in future, we should bring in rest of the immunization registry data and integrate it and use these as a kind of like an ongoing surveillance system for vaccine safety. And similar to Scandinavian countries, our data in BC is quite comprehensive compared to other provinces. I work with teams in Ontario and Quebec and other provinces, and our data is in a pretty decent shape, and we could use it for assessing vaccine safety signals for other vaccines as well as doing these long-term association studies. We may not be able to establish causation given some of the limitations in administrative data sets as well as the time that may have passed, but we could be able to assess association between certain vaccines and certain outcomes while accounting for various confounding and other variables of interest there.
Mm-hmm. Thanks very much. And I know a lot of the limitations are around mobile populations because
Back to you,
I think in the
Monika.
Scandinavian countries they probably have more stable populations over the long term as well.
So just a couple of comments. One on the first question around the long term. So I don't know about 50 years, but we have just started a sort of case control design study for children who are admitted to hospital with APHIS after COVID vaccine, following them out to at least 18 months right now. So we may be, if there's something very soon, but it's not within the first sort of month or so, we may be able to see that. And then I think in terms of the silver lining, I think the other piece for me that's been really amazing with what we've been doing through the special immunization clinic is really the engagement with the adult physicians. And I think, historically, BC's got a very good track record that predates the family immunization clinic at the hospital of assessing children with serious APHIS and being somewhere where pediatricians, public health physicians, family physicians could refer to. I think we've set up a really good infrastructure with the adult clinic side, and I think with increasing adult immunizations, it would be great if we could find a way to sustain that in the long term.
Mm-hmm. And I'll just mention that along with the Vaccine Safety Working Group that I mentioned earlier, we are engaged with the Ministry of Health and the Deputy Provincial Health Officer, Martin Lavoie, in setting up an adult special immunization clinic, whether it's going to be a physical or virtual it's probably not going to be physical kind of place like BC Children's Hospital, but definitely the focus right now is going to be to try to recruit neurologists and allergists especially, as well as general internal medicine, so that there is a place where adults can be seen, especially for the parts of the province where specialty services may be limited within the region. Are there any other questions? Oh, Susan Hollenberg.
I'm going to break the trend. Is this on? So more specific question than the last one. Are there currently studies in Canada through the special vaccine clinics or the Vaccine Evaluation Centre on the use of recombinant shingles vaccine for these kids who are considering live varicella vaccine? using the recombinant product instead of the live product?
Yeah, so that's a great question. I would say the short answer is no. I think there's a lot of interest in doing those studies. Unfortunately, because the vaccine right now isn't approved for use in children, to get the vaccine would require some collaboration with the company who make the vaccine, and then to do any immunologic assessments. Also, right now, those assays are proprietary; they're owned by the company, and so again, that would require some collaboration with the company. My understanding is that there's been relatively limited interest from them to do pediatric studies. I believe as part of their approval with the EMA in Europe, they've had to do some, or they're having to do some pediatric studies, but at least last time, and this was pre-COVID that we had any conversations, there was no intention to do sort of large-scale pediatric studies with that vaccine.
Just I'm aware that there have been some studies, I think recently published, on their use in younger than age 50, HIV positive and hematopoietic stem cell transplants.
Yeah, no, I've seen that. So they've gone into sort of the younger adult sort of immunocompromised population. So I think that the pediatric piece would be would make sense. But right now, I don't know of any sort of planned studies.
Are there any other Slido questions?
There are. I'll go with the most popular one. Our messaging is often that vaccines are safe, which can mean very different things to public health versus the public. When people learn that there are rare, serious risks, this can produce skepticism to our messaging. Should we switch from safe to low risk, or benefits are higher than the risks?
Manish.
Can we ask Ian to respond to that? I mean, I think this is almost a communication question, to be quite honest. So, and I'm not sure I'm the best person. Monika, do you want to take that?
We have Jane Finley here.
Jane, yeah.
She talks to parents all the time.
Yeah, I tell them that vaccines, you have to say they're 99.9999% safe rather than 1 in a million issues. We have to be really, really positive about the vaccine.
And I think historically we've included all kind of APHEs under the safety umbrella. But as you sort of alluded to a little bit in your talk, Monika, you know, the injection site reactions and those sorts of, you know, fevers and general malaise, like we in this room wouldn't consider that a safety issue that would contraindicate future immunization. And I think, you know, when you look at the trials, they're published as safety, tolerability, acceptability. So I think we probably need to sort of distinguish between true safety concerns like the myocarditis and thrombocytosis and things that we're expecting and that are really around tolerability rather than safety.
Yeah.
Yeah,
Expected side effects.
And maybe I'll just say, when you have that parent, it's often a parent, at least in my world, in front of you with these concerns, figuring out, well, what is that safety concern is probably really the first step, you know, and just saying, yeah, all vaccines are safe is not going to be the best way to address it. Are they really concerned about the pain? Or are they concerned about the thrombosis that really occurs in, you know, 1 in 3 million? And then the conversation is going to be very different depending on what that specific safety concern is.
Or are they worried that the government has nanoparticles in vaccines and is monitoring their every move? Yeah.
Yeah, that's going to be a different conversation too.
And I think it's easier to have those conversations, you know, in the clinic setting where that we work in the individual level because you have the time to go through it, or even in the public health clinics. I think what's more challenging, and again, I think this is a communication issue, is, you know, when you're looking for that 3-second soundbite that you have to get up and do on a media interview, or when Bonnie Henry is giving a press briefing, I think that's where it gets more complicated because you don't have the time or space to then go into it in that level of detail. So, but yeah, I'm not. You need to ask a communication expert.
Do we have time for more? Okay. Could the lower risk of myocarditis after the booster dose be because those at intrinsically higher risk, for whatever reason, self-selected out to not receive the booster?
Definitely. We did, Hattie did a great analysis looking at the recurrence among those who did receive subsequent doses, and we didn't see any recurrences. But it's definitely the case that, many individuals will have been advised to defer until there's more information known about the likelihood of recurrence. So I think that those data are still being accumulated. I wanted to mention that in addition to the administrative data analysis that are being done, and Manish, you mentioned a national study, is that the same as the Nat Hawkins group, or is that a separate? Because there are the cardiologists that have also received funding from the Public Health Agency of Canada to look at the follow-up of these myocarditis cases in order to understand better whether, what the long-term outcomes of having experienced myocarditis after these vaccines are. And then Bruce Carlton up at Children's Hospital, and he's a pediatric geneticist, is also assembling a very large set of data, including, interestingly, a lot of US cases, in order to look at whether there is a particular genotype or genetic risk factor for development of myocarditis. And that's why I really think that the Gattaca thing is sort of something of interest for the future. One of the things on my slides was the reference to the Clinical Immunization Safety Assessment Group in the United States, and that's a longstanding group that runs out of academic centers and is funded by CDC as well, trying to look at immunological and genetic correlates, I guess, or risk factors for the occurrence of some of these very, very rare events that probably can be stimulated by an immune response to a vaccine, but clearly not in everybody. They only occur in a very small subset of the population. And so the question is, can those individuals be somehow identified, upfront over time? Perhaps that's going to be possible, and it also might give us a better understanding about what the actual triggers in the immune response are. And it could be a combination of genetics and epigenetics, environmental exposures, all those kinds of things, right?
Yeah, no, exactly. So Bruce is the one we're collaborating with through the SIC. So he's the one who we're sending the saliva samples to for the myocarditis, pericarditis, and the controls for our analysis. And as you say, he's combining it then with these sample sets that he's got from other parts of the world to look for. But he's looking at specific genetic factors. But as you say, some of those, and the genetic factors is, you can't really do anything about that. But what is potentially more exciting is in the blood samples, we're looking at sort of small proteins and other things that may fluctuate over time and also may be modifiable with immunomodulatory drugs in a sort of broad sense of that word, which would be really interesting.
Okay. Julie, did you have anything to add? No? Okay. One more?
All right. Is there any data on COVID-19 vaccine side effects on menstrual cycles? Is this something that will be looked at as a risk factor for giving booster doses?
So so we do have data in Canvas that we're currently analyzing. But there's also been a study, and of course I'm not going to think of who the first author is, out of Alberta. You know what, if there's a way to provide it after the session, I'm happy to sort of provide the link to it. It was actually a really nicely done study because it was it's a well-controlled study, and they did it prospectively, so they were following people forward and assessing sort of hormone levels. Bottom line is they didn't see an effect, if that was what people were wondering, but I'm happy to get the link to the actual publication, and people can read it themselves.
And there have been a few publications actually, and I think that it's been recently reviewed by the Public Health Agency of Canada. And I think that there seems to be it's a very difficult area to study, as you know, because most people don't keep perfect track of their menstrual cycles, and some menstrual cycles are heavier than others. So it's a very, very challenging area to study. But there's work being done not only in Canada, North America, but also in the European countries to take a look at this issue. And it might be, an emerging area of interest. And I think one of the biological plausibility questions has been, is there some, just like we know now that the adenovirus vaccines are associated with thrombotic abnormalities, is there something that might be driving this phenomenon biologically in association with COVID vaccines? So,
and just to add to it clearly, I'm not really an expert on menstruation, but I was involved in a study that Gina Ogilvie and Laurie Brotto were leading at the Women's Health Research Institute, and we did find that, one, it's challenging because of the sort of natural variability of menstrual cycles, but also just the pandemic itself did have an impact on menstrual cycles, and there was changes, particularly in people who had sort of high scores for depression and anxiety. So I think that's going to be an additional challenge in unpicking the pandemic effect from the vaccine effect itself.
Yeah, and maybe I'll just add, I think at a minimum, I hope that this ensures these types of questions get put into randomized controlled clinical trials, because the fact that this was coming up as a signal in our data and we had nothing collected in a controlled setting at that time point just made it virtually impossible to really effectively address it, I think. So at a minimum, going forward, if the drug companies don't start doing that, I'll be advocating quite loudly. So.
Yeah, and I think it's, again, on my international slide, one of the acronyms there was related to better studies of safety in pregnancy. And similarly, the recognition that a lot of this work has not been done in a standardized fashion, that things like, the gestation period and the outcome, the actual outcomes, we really need to have much better processes for, and this is, as you know, a longstanding issue around studying anything in the 70-kilogram male, but ignoring the other half of the population. So better work needs to be done. Okay, so I think that was our final question. So I'd like to thank our panelists very much. Thank you, Julie and Manish and Naveed. And I will just wrap up the meeting. Okay, well, thank you very much for hanging in there. Thank you very much for attending this event. I would like to thank all of our presenters, all of our facilitators. Many of them were members of the planning committee, and the names of the planning committee members are on the back of your program, but they include Lindsay Bowthorpe, Kathy Chilton, Laura Craig, Devon Grayson, who was our timekeeper. Apologies that nobody paid attention to your valiant attempts to keep us on time. Elizabeth Lee, Megan Martin, Stephanie Meyer, and Bhavna Sharma. I'd like to thank them all because they spent several planning hours with us. And we've also had excellent administrative support for the forum. They looked after every aspect of your comfort in the room, including the meals, the program, the Slido, the online questions, and they will follow up with you for an evaluation, which will come tomorrow. And they're in the room, and Daniela Michel, who's our public health manager in our service, Jessica Taylor, who's our operations coordinator, and then Michael Zizek, who's the event consultant from Two Sparrows Media, kept everything on track. Thank you. And I'd like to thank our excellent AV team as well, because we had a bit of a better experience this year than last year. Thank you very much. It was excellent. Everything worked, including our remote participants. I'd like to just thank the program committee for the fantastic program, because I think we had a great balance of science information and practice-based information. So I hope it was relevant to everybody in the room. And I'd like to also thank the BC Immunization Committee for funding this event. That's the source of the funding each year, and we just yesterday submitted the proposal for funding for the forum next year. In times gone by, this used to be a 2-day event with morning plenaries and workshops in the afternoon, but we have gone to a 1-day event, I guess, just to spare people the time. But I think that just knowing how today's program went, I know that everybody feels that there's a lot of information compressed into a very short time. So we will be considering how best to deliver the program in coming years. And it's very important for you to get your evaluation suggestions into us in response to that message that you receive tomorrow, to give us ideas about what sort of content you want next year, what topics would you like covered, and what type of forum or format of the program you would like as well. I've been very honored to be part of this event, and it's been wonderful to be with colleagues like you in the room. I so appreciate the immunization community because I think we are a very special group, and it's been wonderful to see everybody in person. And the forum will be archived online, so each speaker will be asked to authorize the posting of their slide set, and I think we also have the facility to post the live stream, archive it online, so that for people, especially if you participated online and weren't in the room for the whole day, you'll be able to access that information subsequently. And I think those are all the remarks. Oh, somebody left a gray scarf. It's fuzzy and very nice. And so if you are that person who don't want to go home without your scarf, Jessica Taylor had it. And so please don't leave without it. So thank you very much, everybody. And we hope to see you again next year.